Now, this course is titled Fundamentals of Genetics. Okay, and it's a 1+1 course, just like what you had in cytogenetics. The number is GPB 211. Okay, and the contents of this course must have been covered by now in your 12th standard, as well as in your 10th standard as well, isn't it? Yes or no? The contents of this course must have been covered both in your 10th standard as well as 12th standard. So whatever we deal in this course is going to be a kind of revision. In some aspects, we'll go in depth. Otherwise, in majority of the cases, it's going to be a kind of revisiting these concepts. Okay. And we will primarily discuss, or to begin with, Mendelian genetics. Okay, we'll basically deal with or start with Mendelian genetics. So, how do you define genetics? Anyone? How do we define genetics? What is it actually?
Study of inheritance and variations.
Inheritance and variation. What is inheritance?
Transfer of traits from parental to progeny, sir.
Okay, so it's basically transmission of traits,
Yes, sir.
passage of traits from one generation to the other. It's basically passage of characters or traits from one generation to the other, or from parents to the progeny. That is inheritance. Now, what is variation?
The degree of difference found in progeny from the parents, sir.
So, the differences that you see for a character, the differences that you see for a character. Now, when I say differences that you see for a character, let us say mother is brown-eyed, father is black-eyed, they have children. Do you think all the children will have brown-eyed? Or will they have black? Or will they have differences, something different from brown and black? Is there a possibility? That means to say that you would see some kind of differences for a character among progeny or among children from a pair of parents. Is that clear? Differences that you observe among or between progeny from the same pair of parents. Okay? Variation could be hereditary or environmental. Variation could be either hereditary or environmental. Well, Hereditary variations primarily arise because of recombination. Because of recombination between alleles from the two parents. So what happens is you have parent A, then you have parent B, then you have the F1. In the F1, you have the chromosomes of A and the chromosomes of B. You have studied cytogenetics, right? You have studied cytogenetics, yes or no?
Yes, sir.
Yeah, where are the genes located?
Sir, on chromosome, sir.
Yeah, you have genes located on the chromosome. Can you see this? Is it visible or do you want me to write a bit above? Is it visible?
Yes, sir.
Okay. So, now we have something called as maternal chromosomes and paternal chromosomes. Okay. Now, please remember, any organism, any sexually reproducing organism will have two sets of chromosomes. Any sexually reproducing organism would have or would carry two sets of chromosomes in a cell. That's what you've studied in cytogenetics. So, all sexually reproducing organisms primarily come from or develop from a single cell. How was the zygote formed? In all sexually reproducing organisms,
By fertilization, sir.
yeah, by fertilization. Let me repeat. In all sexually reproducing organisms, any organism that you see, any plant, any insect, any human being, they all primarily start from a single cell. The single cell is zygote. And this zygote results from fusion of the female and the male gamete. And we know gametes carry only half the chromosome as that of the somatic cell. So the number of chromosomes that is there in the female gamete is haploid, the male gamete is haploid, two haploids fuse together to get a diploid zygote. Are you convinced? Gametes are haploids. The female gamete is N, the male gamete is N. These two fuse to give rise to the diploid. That means to say that the zygote has two sets of chromosomes. One set of chromosomes that we refer to as the maternal set, another set is the paternal set. The zygote will carry two sets of chromosomes. The zygote undergoes mitosis. The zygote undergoes mitosis. The descendant cells, after mitosis, you'll get two cells. Both the descendant cells will undergo mitosis. Further, these descendant cells will undergo mitosis. Okay? And one cell becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, and so on and so forth. And that is what we are. We are made of a few billion cells. A few billion, BILLION, a few billion cells. Did you get it? Did you get it?
Yes, sir.
Okay, now because it is mitosis, whatever the genetic constitution was there here, whatever combination of genes were there in the cell will be the same here as well. Or you take it to any level, any thousands of cells or millions of cells, all the million cells are genetically similar. Why? Why are all the cells similar?
Because equational division.
Simple. Right from the zygote until the formation of the adult, where we are made up of billions of cells, all the billions of cells have genetically identical material. When I say material, the genes are same across all the cells. That is because of mitosis. Mitosis is an equational division. Clear? Is that clear? Then, some of the cells, now you have millions of cells, some of the cells will undergo the reproductive phase. Not all. What are those? These tissues are called gonads. There, meiosis will take place. Is that clear? Now, in this, for example, you have a cell. We told you there are one set of chromosomes that have come from the female, the other set from the male. Let us say this is a set coming from the female, this is a set coming from the male. Now, the male and female can differ for a number of characters. Of course, these characters are governed by genes. These genes are located on the chromosome. These genes are located on the chromosome. Now, when this cell enters meiosis, pairing takes place between pairs of chromosomes. Pairing takes place between pairs of chromosomes. Then crossing over takes place. Crossing over will lead to different combination of genes. Is that clear? Will lead to different combination of genes. That combination of genes will now result in formation of gametes. Again, it could be male or female. Whether if this is an individual is female, then you will get female gametes. If this individual is male, then male gametes. Now these again will fuse to form a zygote. Now this zygote now will carry again two sets of chromosomes, but in terms of the trait expression, this individual could be different from here. Well, or if there are two children are not identical. For example, there are a few Gavi, on the Erdimshapa, I have some instructions to be passed to our staff. I'll get back to the class. Just a minute. Okay. So if a few cells or tissues undergo meiosis, and we have two events during meiosis. One is random arrangement of paired chromosomes on the equatorial plate. One is random arrangement of paired chromosomes on the equatorial plate. And the second event is crossing over between linked genes. You have genes, if there are a number of genes on the chromosome, there can be a crossing over here. During PAC18, there is a process called as crossing over. During anaphase 1, there is random orientation of chromosomes on the metaphase plate. We say that is bivalence. Bivalents are arranged on the equatorial plate at the end of metaphase 1. During anaphase 1, paired chromosomes separate, paired chromosomes disjoin, and they migrate to opposite poles. I think you have studied this in meiosis. Yes or no?
Yes, sir.
Yeah. So there are two events during meiosis 1. One is during PAC18 where there is crossing over between linked genes. Crossing over between linked genes. Where are these genes? They are present on the maternal and paternal chromosomes. They are present on the maternal and paternal chromosomes. Crossing over between maternal and paternal genes results in formation of new combination of genes. This new combination of genes will lead to different trait expression. Is that clear? Is it clear?
Yes, sir.
Second, there's
Yes,
another
sir.
event that takes place. Yes?
Yes,
There's
sir.
another, yeah, Eli. Eli.
ಸರಿ ಇಲ್ಲ ಏನಿಲ್ಲ ಸರಿ.
There's another event that takes place during meiosis. One is crossing over. The other is, say for example, you have one chromosome here, the other chromosome here. Now, on this chromosome can move here, this also can move here, this can move here. Whereas this, and this, of course, you have 10 chromosomes here, 5 chromosomes move to opposite poles. Either of the two could move to one of the poles. Is that clear? When we have two chromosomes here, this could come here or go there, depending upon how this pair of chromosomes orients themselves on. Now, if you now consider this is the maternal chromosome, this is the paternal chromosome. Now, this bivalent, they are paired chromosomes now. Maternal and paternal chromosome will pair. They align on the equatorial plate. They can align either like this or like this. In this sense, let us say this is paternal, this is maternal, this is paternal, this is maternal, this is maternal. So you have different combinations of maternal and paternal chromosomes moving to opposite poles. Now, this movement of different maternal and paternal chromosomes to one pole results in a new combination of traits in the progeny. So that is the reason why in a family of two brothers or in a family of two sisters, both the brothers are not identical. Both of them are born to the same parents. Two sisters are not alike, but they are born to the same parents. Why? Because of these two events that take place during meiosis. Okay? Therefore, two sisters are not identical genetically. Two brothers are not identical genetically. When two brothers or two sisters, when you can imagine if there are 10 siblings, all the 10 siblings are not genetically similar. That is variation. Is that clear? What kind of variation is that? That is hereditary variation. That is called as hereditary variation. Apart from hereditary variation, you also have something called as environmental variation. Environmental variation. Well, what is this environmental variation? If Gavi Siddhaya is studying in GKVK, He scores in agronomy 96, soil science 96, genetics 96, or whatever it is. You take the same Gavi Siddhaya to Dharwad. Will he score 96 in all these subjects? Maybe he could score much more. The environment at Dharwad could be more congenial for to score much higher. Now, this is environment. Is it clear? So, the environment could influence the expression of a character. What is the character that we are talking about here? Percentage of mark scores and KVK is one environment. Dharwad is another environment. Shimoga could be the third environment. Is it clear? Is it clear?
Yes.
Okay. So now we have understood what genetics is. Genetics primarily is a branch of science that deals with inheritance and variation. We have seen what inheritance is. Inheritance is nothing but passage of traits. Passage of traits from one generation to the other. Or passage of traits from parents to offsprings. Or transmission of traits from parents to offsprings. That is AMB 0089. Please remember, I'm going to make note of the ID numbers along with their names. From whom there is disturbances. You should be aware that you cannot be disturbing the class. '89 Preeti or someone, I don't know. Could you please mute your voice? Gavi Siddhaya.
Return off your mic.
Yaru AMB 0089.
Return anta sir.
Pritama.
Return sir.
Sir.
Okay. So, in this class, we'll start about Grigor Johann Mendel. How many of you have heard this name and how many of you have not? Please make note of the spellings. Gregor, Grigor, Johann, Johann, Mendel. Grigor Johann Mendel. Let me tell you, he was not a student of biology. He was not a student of biology. His specialization is not genetics. He was primarily a priest in a church. He was a priest in a church, and he did not know biology to a very great extent. And he proposed his laws somewhere in the year 1865. In 1865, nothing was known about who discovered chromosomes?
Waldeyer, sir.
Yeah, Waldeyer. Which year?
78, sir.
What is this year when Grigor Johann Mendel proposed his theory?
65.
1865. That means to say that Mendel had no clue about chromosomes. Is that clear? Is that clear?
Yes, sir.
Mendel did not know anything about chromosomes. Okay. Did Mendel know anything about mitosis? When did Fleming describe mitosis? Any idea? When was mitosis described? What is the literal meaning of mitosis? What is mitos?
Sir, equational division.
No, what is mitos? Mitos is threads, division of threads. Okay, chromosomes were actually visible as thread-like structures. Okay, and they saw that the threads were dividing. So this is nothing but the division of the threads. Did you get it? Is it clear? Well, when did Fleming propose mitosis? It was in the year 1894. Then meiosis. When was meiosis described? Who described meiosis?
Sir.
Who described meiosis?
Farmer and Murray.
Farmer and Murray described meiosis. When was that? 1882 is mitosis, 1894 is meiosis. And please see all these events. All these events took place after Mendel proposed his rules or concepts of inheritance. That means to say that Mendel had no idea about mitosis. Mendel had no idea of meiosis. Mendel did not know what were the factors and where were the factors sitting. He called them as factors which we now know as genes. Is that clear? So Mendel had no clue, but he gave to the world a concept called as rules of inheritance or laws of inheritance. And you know when Mendel was born? When was Mendel born? This year, July 20th, we'll be celebrating completion of 200 years of Mendel. This year, we'll be celebrating 200 years of Grigor Johann Mendel. Okay. How many years from 1965? 1865. 157 years. Mendel's findings is now 157 years old. Did you get it? 157 years old. There is nothing that can contradict the rules of Mendel. Is it clear? Is that clear? That means to say that whatever Mendel proposed as rules of inheritance holds good even today. Maybe it will hold good for some more years, some more decades, some more centuries. We do not know. What are these rules? What are these rules that he proposed? How many rules did he propose?
3 rules.
What are those 2 rules?
Law of dominance, law of segregation, and law of independent assortment.
So how many?
3, sir.
Law of dominance, law of segregation, and law of independent assortment. That's what you said, right? Are there any other laws? Is there any other law? This is wrong. Is that clear? Law of segregation, yes. Law of independent assortment, yes. Is dominance called as uniformity. Law of uniformity.
Yes,
sir.
sir.
Okay. So henceforth, please don't say law of dominance. Clear? You do not have something called the law of dominance. Law of uniformity, law of segregation, law of independent assortment. Is that clear? Now, can you write?
Sir, you are not audible.
Okay. Am I audible now?
Yes, sir.
Okay.
Yes, sir.
Now, even before we get into the rules or laws, there are certain terminologies that we may have to get acquainted with to understand these laws. There are certain terminologies. What are these terminologies? For example, in genetics, there are some unwritten conditions. Okay? What are they as? Unwritten conditions or unwritten rules ಏನಂತಾರೆ? One of the
Postulate.
yes,
It's postulate, sir.
postulates. They're basically called as conventions. There are certain conventions. What are these conventions? For example, in India, when you're driving a vehicle, you have to drive on your left side, isn't it? Yes or no? You drive on your left or right?
Left, sir.
Yeah, that's a convention. Now, it's not written anywhere. There's no rule. but you'll have to drive on your left. Is that clear? When you write the first parent here, when I write P1 is parent 1. P1 stands for parent 1. P2 stands for parent 2. Is that clear? Now the first parent is always what? Is always female. The following parent is always male. That is how we symbolize female and male. There is a symbol for female. There is a symbol for what is the symbol? What is that symbol? What is the symbol?
Symbols of male and female gametes, sir.
And that is symbol is fine, but yeah, what does it depict?
Gender.
Huh?
Gender of the individual, sir.
No, gender is fine. What does this symbolize? How many girls are there in this group? Now, of course, these days nobody uses a mirror. These days nobody uses a mirror. Our Android mobile phones have replaced several things, isn't it? No, this is the mirror part. This is the holder part. And maybe 15 years back, if you open any lady's wallet or purse, you'll find a mirror in their bags. Okay, that is why this symbolizes female. What about this one? What does this symbolize? This is an arrow. This is a shield. Males are often expected to be brave and be involved in protecting themselves, fighting for something, using a shield and an arrow. This symbolizes male, this symbolizes female. Is it clear? So we'll be using this symbols in genetics quite often, or even till the end of this semester. Okay. The progeny derived from this cross. Now, this simply implies that a cross is made. Now, in plants. Now, the moment we say P1 is female, you don't have female plants. Did you get it? Do we have female plants? Exclusively female plants? Idea.
No, sir.
Yes, sir, in cucumber, sir.
In cucumber, amele. Eli, inna.
Papaya, it's there.
Yeah, papaya, date. Very few. Otherwise, you would have both male and female reproductive units in the same flower. That's why we call them as bisexual. Most of our crop species are bisexual. Or we also call them as hermaphrodites. Hermaphrodites. That means to say that you cannot have a female plant or a male plant. So you'll have to designate the plant as male or female. Did you get it? How do you designate this? How do you designate this fellow as female? remove the anthers, remove the male reproductive unit. When you remove the male reproductive unit from the flower becomes female. Did you get it? And that is how you designate this as female, this as male. Okay, so when you have removed the anthers from here, or when you remove the male part, you get pollen from here and dust it onto the emasculated stigma. Now, emasculation is nothing but removal of the male parts. Emasculation, masculine. You are removing the male. That's why it's called emasculation. You remove the male parts, so you make this female. You bring pollen from a desired male, pollinate the stigma. You have studied all this right in cytogenetics. Well, when you harvest seeds from this, what have you done? You have allowed the pollen to fuse with the eggs here. Hybrid seeds are formed on the female parent. That is why this is called as the seed parent. And this is called as the pollen parent. Any difficulty in understanding? Any queries? Please feel free to ask. Anything? Can I go ahead?
Yes, sir.
Okay. Now, when you make a cross between these two, what do we get? So, you have designated this as female parent by emasculating the flowers, removing the male parts. You bring the male part in the form of pollen. And please remember here, the female gametes are stationary. The female gametes are stationary. The male gametes are mobile. The male gametes are mobile in plants. If I say the female gametes are stationary, that would be wrong. I need to tell that the female plants, when you plant something, that will stay there only, it will not move. But how does it get the male? The male will come in the form of pollen, either through wind, either through insect. Did you get it? The male gametes produced by this can be carried, but the female gamete cannot be carried here. So therefore, this is called as the seed parent, because seed is nothing but the F1. So when you make a cross, you get the F1 hybrid. What is F1? This F1 actually means first filial. First, this is first. This is filial. First filial. Is it clear? Is it clear? The first parent that you write is the male, sorry, female. The second parent that you write, the following parent is nothing but the male. When you make a cross between them, you get the F1. The F1 is regarded as the hybrid. The generation is referred to the first filial. Well, if the F1 is crossed either to A or B, what is it called as?
Back cross.
Back cross. This is called as the back cross. Okay? Now, instead of making A as female, if I make B as female and A as male, you get an F1. What is this cross called as?
Reciprocal cross.
Yeah. Now, A into B and B into A are a set of reciprocal crosses. A set of reciprocal crosses. No, reciprocal crosses doesn't mean only one. It has no meaning. What is reciprocal cross? A into B, you derive an F1. B into A, you derive an F1. Now, these are products of a set of reciprocal crosses. In one of the cross, you have made A as female, B as male. In the next cross, you have made B as female, A as male. In both the cases, you have the F1 hybrids. Okay, these two F1 hybrids are products of a set of reciprocal crosses. Is that clear? Then why do we perform reciprocal crosses? Why do we do this? Why do we perform or make reciprocal crosses and try to study inheritance? Kindly make a note. A set of reciprocal crosses are made to determine if the trait is under the influence of nuclear genes or cytoplasmic genes. To determine whether the trait is under the influence of nuclear genes or cytoplasmic genes. If the trait is under the influence, the trait under consideration is under the influence of nuclear genes, then there will be no difference, there will be no significant difference in the phenotypic expression of these two F1 hybrids. Is that clear? If the trait is under the influence of nuclear genes, now please remember, if you have a cell, within the cell you have the nucleus, there is the cytoplasm. There is the nucleus, there is the cytoplasm. In the nucleus you have the chromosomes. In the cytoplasm, where do you have genetic material? You have the mitochondria and the chloroplast. Some DNA is present in the mitochondria and the chloroplast as well. Is that clear?
Yes, sir.
Is that clear?
Yes, sir.
Now, if the trait is under the influence of nuclear genes, if the trait is under the influence of the character is under the influence of nuclear genes, the two F1s would be identical in their phenotypic expression. If the trait is under the influence of mitochondrial genes or cytoplasmic genes, then the trait could differ. These two F1s could differ. And the trait expression of the F1 will depend on the female parent. The trait expression could depend upon the female parent phenotype. Is that clear? If there is no reciprocal cross differences, then we assume that the character is under the influence of nuclear genes. If there is significant differences in the F1 hybrid phenotypic expression from a set of reciprocal crosses, then we presume that the trait is under the influence of cytoplasmic genes. Clear? Is it clear?
Yes, sir.
Okay, that is the purpose of making reciprocal crosses. What is the purpose of making back crosses? What is the purpose of making back crosses?
So to identify the unknown genotype of the plant, sir.
Okay. That is what you have learned all these years, isn't it? Okay. Now, a test cross is specifically done. A test cross is specifically done to assist a geneticist, a person who's working on genetics, to assist a geneticist to phenotype the gametes. To phenotype the gametes. Can you phenotype the gametes actually? Can we really phenotype the gametes?
Genotype, sir.
Huh?
Genotype, sir.
We don't. Okay, you said genotype, you determine the genotype. But I'm telling you, the very purpose of performing a back cross or a test cross is to phenotype the gametes. Did you get it? We'll see how we'll phenotype the gametes. Truly speaking, When you hold a egg, when you hold a pollen, can you tell what kind of gene it carries?
No, sir.
You cannot, isn't it? You can. But the progeny derived from a backcross or a test cross will enable a geneticist to phenotype the gametes. Okay? You can predict the phenotype of the gamete. We'll discuss that a bit later. Okay? That's the purpose of backcross and test cross. Okay. Now we have understood what reciprocal cross is. Okay? What a backcross is, what P1, P2, F1 is, isn't it? Now we'll come to what a gene is. Who gave the concept of gene?
Johansson. Johansson.
No, Johansson gave the concept of genotype and phenotype. William Bateson. Anyway, what is a gene? How do you define a gene?
Gene is a unit of heredity, sir.
Unit of heredity. Anything else? That's a very, very primitive definition. A very primitive definition. What is a gene?
It is the combination of DNA molecules.
Combination of DNA molecules, is it? How many DNA molecules can you expect to get from a chromosome? Who was your cytogenetics teacher? ಯಾರು cytogen ಯಾರು ಕಲಿಸಿದ್ರು?
Sir, gene is a sequence of nucleotide present at a particular locus of chromosome.
ಸರ್ ಮೈಕ್ ಆಫ್ ಆಗಿದೆ ಸರ್.
Okay. I don't know. ಯಾರು ಮಾಡಿದ್ದು?
Sir, gene is the small sequences of nucleotide which contain specific information, sir.
Yeah, information is fine. But what exactly does the information do? What is the information for? So, a gene is nothing but a segment of DNA. Okay? Okay. That DNA will undergo transcription, then translation. Clear? In some cases, product of transcription itself is the end product. What is the end product of transcription? You will get different kinds of RNAs. Okay? Messenger RNA will go on to become a polypeptide. Whereas you have other RNAs like ribosomal RNA, transfer RNA, heteronuclear RNA, small nuclear RNA, small interfering RNAs. So they are products of a gene. That means to say that a segment of a DNA that codes either for different kinds of RNAs or a protein or a polypeptide. Is that clear? Now, this polypeptide or protein is responsible for the expression of a phenotype. Say, for example, a protein or a polypeptide would be required for a particular flower color, a particular seed coat color. Is that clear? Then what is an allele? What is an allele?
Alternative form of gene is allele, sir.
Alternate form of a gene is an allele. Okay? alternate form of a gene is an allele. The most commonly found allele is called as what? No, we have, okay, you have gene A. Let us say we have gene A. Now it has alternate form. What are the alternate forms? We can call them as A1, A2, A3, A4. These are the alternate forms. For example, in terms of color, let us say you have rose. Among rose flowers, you have different colors. You have red, deep red, pink, white, yellow. Now, this is a gene controlling flower color in rose. A1 controls, let us say, deep red. A2 controls, let us say, red. A3 controls pink, dark pink. A4 controls light pink. A5 controls yellow. A6 controls white. Did you get it? These are the alternate forms of a gene. Alternate forms of a gene are called as alleles. Is that clear? Is that clear? So
Yes.
we have understood what a gene is, what an allele is. We have understood what reciprocal crosses are, what back crosses are, what F1 is, what parents are, and things like that. Any doubts in the aspects that we have covered so far?
No, sir.
Okay. Well, whether it is online or offline, please remember now itself that you may have to consult some important books. Okay. Well, most of you probably should now be having access to Fundamentals of Genetics by B.D. Singh. Fundamentals of Genetics by B.D. Singh. Please consult that book. But students who want to know a little more on genetics, who have keen interest in genetics, now for all the students in the class, Fundamentals of Genetics by B.D. Singh is fine. Okay? Or Genetics by P.K. Gupta is fine. Genetics by P.K. Gupta is good. But some students would be there who want to learn a little more about genetics. For them, Principles of Genetics by Robert H. Trammon. Robert H. Trammon. Then others, the title is the same. Snustad. S-N-U-S-T-A-D and Simmons. Read these two books. Now, I don't suggest everyone, it will be wrong on my part because you have several subjects. Those are very keen to learn the science of genetics in a much better way. For them, Principles of Genetics by Robert H. Trammon and Snustad and Simmons. For the entire class, Fundamentals of Genetics by B.D. Singh or simply Genetics by P.K. Gupta would do. Is that clear? Another word of caution here. Genetics is not a subject that is very tough. Okay, I've seen the question. I'll answer you. I'll get back to the chat box a little later. Now, one thing is, genetics is not at all tough. It's a very, very easy subject, provided you involve yourselves in the class. If you do not involve in the class, yes, it could be a bit difficult. I'll let you know as we go ahead. So while writing product of crossing, we write F1 generation, F2 generation. What is the full form of the letter F? F is filial. Filial. Is that clear? I think 32 Banu. Okay, right, fine. Shall we close for the day? Gavi Siddhaya? Shall we close? Do you have any other doubts?
No, sir.
Okay then. Okay. We'll meet. We have a class next Thursday, right? A section class ಗೆ Thursday ಅನ್ಸುತ್ತೆ. We'll meet. Okay? Okay then. Bye.
Thank you.
Right. Thank you.
Recording stop.